series presented by Women in Cotton, which we spotlight women in the industry for them to share their industry knowledge with participants. Today, we are lucky to have Dr. Colleen MacMillan joining us to discuss her experience in cotton research. Before we begin, we just want to briefly go over a few items and rules prior to our discussion. So our rules for today will be, so please be kind to yourself and to others and aim to be the best version of yourself. Please be thoughtful, responsible, and bring a positive energy to this discussion. Listen with openness and curiosity, and please remember as well to mute yourself when you're not speaking. We welcome you to share your stories, focusing on positive outcomes and using generic descriptions without naming names. From time to time, topics do arise in these conversations where you may need to seek special support. So please do reach out to your HR department if that is the case. Be prepared to think laterally. We are all about diversity, inclusion, and innovation in Women in Cotton and our Women in Action events. So do be prepared to think out of the box to bring out extraordinary solutions. And then finally, acknowledge that as women and men, we can and should make a difference to everyone's life at work. So now I would like to introduce Dr. Colleen MacMillan, who I mentioned will be presenting to us today. Dr. MacMillan is a plant molecular biologist researching cotton. Based at Black Mountain, Canberra, Australia, she is the Cotton Fibre Team Leader in the Cotton Biotechnology Group at Commonwealth Scientific and Industrial Research Organization, Agriculture and Food. Colleen specializes in biotechnology-based research to help create solutions for sustainable fiber-based industries. Currently, this includes a range of collaborative research areas such as CSIRO's Future Science Platforms, SynBio Novel Fibres Project, and Fiber Cell Wall Synthesis and Function Projects, and CSIRO Agriculture and Food, and with other CSIRO and external collaborators in Australia and internationally. In addition to science, Colleen has a strong commitment to inclusion and diversity for science and is the inaugural Inclusion of Diversity Executive Lead for CSIRO Agriculture and Food. She has been very active and a foundational member of Women in Cotton since the very beginning. So Colleen, thank you very much for taking the time to speak with us today, and we look forward to your presentation. I'll hand it over to you.
Thanks so much, Katie, and thanks very much, Amy and Katie, for the invitation to talk and share and engage today. I'm pretty excited to be here with you all. I'd like to acknowledge the traditional owners of the land that I am presenting on today from Australia, and that is the Ngunnawal and Ngambri people, and I pay my respect to their elders past and present. I also extend my respect to any First Nations people who are watching or connected to this presentation, and also to everyone from dialing in from wherever you are. A story about a very interesting First Nation young woman, a Kamilaroi woman. Her name is Thelma Plum. She's a singer, and I came across her last year. She had an inaugural album as a First Nations woman in Australia called Better in Black. And I was reflecting on that album and the title song Better in Black as I was preparing for this presentation, because the way I've been doing science is not the stereotypical way of doing science. And when I first started realizing I don't fit the mold of the standard stereotypical scientist, it was a challenge. But hearing a song like that, Better in Black, it's very releasing for me, but also for anyone on this call, to be yourself, do things your own way, and everyone wins that way. So if you have a chance to listen to Thelma Plum's song, Better in Black, I encourage you to, but there is a language warning in there too, just in case there's young people around. So it's such a pleasure to be with you here. And so the theme throughout my talk today, and as I engage with you, is around different ways of doing science. And I'm going to actually pull out the themes, not explicitly, but the themes of individual leadership, the theme of co-leadership and collaboration, which is very important, but it's not the norm in science. So I hope that you'll pick up on that throughout the science that I'm going to present. So here we go. I'm going to share my screen now. All right, if someone can be so kind as to give me a thumbs up if they can see my screen. Yep, beautiful, thank you very much. So very nice to be here. I am working for CSIRO, Australia's National Science Agency. We have about 5,000 people across about 50 sites across Australia. And it's a $1 billion organisation, but about 40% of that revenue comes from external sources. We have 7 science lines of business. So I'll talk a bit more about agriculture and food, which is one major line of business, but we also have land and water, oceans and atmosphere, health and biosecurity, manufacturing, mineral resources and energy, plus a lot of other units as well. So in the National Science Agency, one of our units is Agriculture and Food Business Unit. We generally have about 1,000 staff and affiliates, including students and honorary fellows and casual workers, across at least 16 sites that you can see there on the Australian map around the country. So I'm presenting here from Canberra, literally in the Synergy building that you can see there at the Canberra Science and Innovation Park, and I'm based in the Cotton Biotechnology Group. So a little bit more about agriculture and food. We do very broad-ranging research. We research traits, lots of different types of traits in crops, future crops, farming systems, livestock and aquaculture, sustainability and food. And in our food program, we even have a pilot plant for food production. So it's a very interesting and well-connected, truly agriculture and food business unit. So our cotton research spans breeding, and that's the main piece that the globe benefits from in terms of CSIRO and cotton research, biotechnology, agronomy, and textiles. So a lot of our biotechnology work feeds into our breeding work. And in the map there, you can see, let me just grab my laser pointer, here at Millvale, that's where Australia's cotton breeders for CSIRO are based, and that's where they've been doing at least 50 years of extraordinary cotton breeding. And we are very closely connected with that group, as you will see. So back to the Cotton Biotechnology Group here in Canberra. This is our Cotton Biotechnology Group. It's a wonderful team to be, a group to be in, and I want to point out some people that I have been co-leading science with and collaborating with over several years, and some of that work you will hear today. I'll point out Dr. Filomena Petalino, who is one of Australia's best cell wall biologists, Dr. Chao-Ching Lee, Dr. Dina Yulia, Lizette Perez, Dr. Danny Llewellyn, Dr. Viv Rowland, and I am there as well. And I collaborate with the rest of the group as well, but I wanted to specifically call out those people that I collaborate very closely with in the fibre and fibre quality space. So getting into some science here. So I think that the cotton plant has truly extraordinary biology crossed with human endeavour. And the more we talk about this, the more we can think about the past and shape where we're going into the future. So the cotton plant is the world's largest plant fibre commodity for textiles. We know that is very clear. Number one, it's a plant-grown fibre. It's renewable by definition because it uses these things, these green things called, solar panels, called leaves that can harvest sunlight in a very clever way. Cotton fibres are biodegradable and human bred. So I'm going to touch on a few of those. So one of the things that I fell in love with when I left forestry, the forestry sector in Australia, and came to do research in cotton more than 13 years ago, my jaw dropped when I when I learnt that the cotton fibre that we weave into textiles, each of those fibres is a single cell. It's more than 3 centimetres long, and it's a cell that you can pull off a bowl and hold it in your hand. You don't need a microscope or anything. You can just hold a single cell in your hand. The biology to achieve that is really, truly extraordinary. We must always remember that plant-based fibres, so if you think about cotton, linen, hemp, bamboo, they are incredible because they harness photosynthesis to capture energy, fix carbon, and produce amazing fibres. Trees are also obviously a really good example of that. So a little bit about the fibre and the unique biology that we've been researching. So on the day of flowering, when that beautiful flower opens on the cotton plant, we see the following. So on the vertical axis, the y-axis, we can see fibre length here in centimetres. And across the x-axis, so across the horizontal axis, you can see the days after that flower has opened. And on that very first day when the flower opens, on the surface of the seed, there are about 30,000 of these tiny little cells that start to balloon out, and they start to elongate over 2 to 3 weeks. So in green there, you can see that. And what I've drawn here is the wall of that ballooning cell. That wall is very, very soft and elastic. So it allows that cell to elongate in a very special way. There are beautiful molecules within that wall that are structured in a certain way to allow that ballooning. So we've been going deep into the molecular structure of that to understand that and then harness it for the work that we do. At about the 3-week mark here, we see that inside of that beautiful soft balloon cell is a very thick wall that starts to be deposited, and that occurs up to about the 2-month mark, up to the 40-day mark, and it just keeps depositing more and more of this highly cellulosic material. On the right there, you can see a little cross-section of all of those long chains of cellulose that I've drawn there to just indicate how thick it is. And then the cell dies, and then the bowl opens, and it presents the fibre. We don't have to ret or extract out of the plant. It presents the fibre that we gin. And that's one of the beautiful things about the cotton plant. We don't have to use water to get the fibre out. And I love that. What we've been researching in particular is the DNA through to material properties. So not just individual DNA molecules, but across the entire genome. So there would be 30 to 60,000 genes and gene classes that we look at, and how those genes have variants, but all to this aim. So if we take a fibre and we do a cross section, we see here painted in blue that thick secondary cell wall. Now we've measured what is in that wall. It's more than 94% cellulose. There's virtually no lignin, so it's a non-lignocellulosic wall. It's a highly cellulosic wall, which is extraordinary, really, truly extraordinary in terms of then being able to produce that high tensile strength. And that is the basis of cotton being grown as a commodity worldwide. There's also some of those hemicelluloses I was talking about before that allows that ballooning. And we actually know a lot about what is in those classes of hemicelluloses and even some proteins actually, which change the material properties. So in particular cellulose, so for those who haven't yet had the chance to fall in love with cellulose, cellulose is this. It is so purely simple. It's a very long linear chain of sugar, glucose units, linked carbon 1 to carbon 4. Just a simple long chain. The other thing about the cotton plant, not in the fibre, but in the stem, I see cotton as a bit of a tree, given my forestry background, but we've also measured the lignification in cotton, and cotton does very unusual things with lignins. So these are the types of lignins that we've managed to measure using nuclear magnetic resonance with our partners in the US. So what we've done is we've been looking at all the genes from that moment of their fibres elongating to making that thick wall, thousands of genes and their variants to see what is controlling that pathway to make these amazing materials. So here's one of our biggest papers that we published. Yeah, if you're interested in exploring that. So not only the genes that form the structure, but the genes that are like the CEOs, they're called transcription factors, and they will turn on the entire pathway and say, now's the time to turn on this, and please go ahead, all these departments, please make X, Y, and Z and do it in this way. So that's my particular interest, is those transcription factors, who they are, how they work, and who they turn on. So also a little bit more about the unique evolution of cotton and domestication. So the Gossypium species, to which hirsutum belongs to, that species, that genus itself evolved about 10 million years ago with about 50 species, of which there have been about 4 where there have been particular independent human domestication events, and that has occurred between 5 to 10,000 years ago. So I've painted those 4 regions here. So when we come to Australia, where I'm presenting on, I'd like to take you down into the Australian space and talk a little bit about what we've been doing in Australia. Before I jump into that, in Australia, it's very normal for the whole plant to be used, not just the fibre, but also the seed for oil and various other things, and the stem and the leaves. We can talk about that later if you like. So in Australia, when we look at cotton, in 1788, cotton seed first arrived in Australia via the first fleet via Brazil, and there was no cotton really planted. But the modern era of cotton in Australia really started
bedside table.
1960s. So there were 2 American cotton farming families and a European cotton breeder who arrived and started doing some breeding. But in 1971, leading to 72, the National Science Organization, CSIRO, made a decision to bring together its cotton breeding unit together in that Millvale site 50 years ago, and we're constantly seeing innovation, improvement, up with the latest technology, pushing the boundaries of where science can go, and a beautiful partnership with the entire industry, including research, development, and extension. So it's a very pivotal moment there. In 1985, the first bacterial blight-resistant variety was released, and that reduced insect damage. In 1991, a variety was released to improve resistance to the fungus Verticillium wilt. In 1996, the first GM cotton was released in Australia with InGuard, which had insecticidal resistance. 2003, Bolgard 2, which had insecticidal resistance as well as protection, the inbuilt protection against traits. Bolgard 3 had also insect resistance trait released, and 95% of Australian crop was then GM at that point in time. So where do we go from here in terms of the research we've been doing? I will call out CSIRO's cotton breeders here as being absolutely standing next to the best breeders of cotton in the world. They really are extraordinary, and it's a real privilege to work with them. Now, coming to fibre. So global fibre production since 1990 has started to see a very almost exponential increase. So if we look at the bottom bars, so the vertical axis is million tonnes, and the horizontal axis is time since 1980 to 2020. So since 1990, we can see the bottom bars here are the natural fibres, so cotton, wool, etc. That's been on a gentle increase, but what we've really seen is that the synthetics in blue have had a massive increase in global fibre production, largely polyesters and a few others. The man-made cellulosics have also seen an increase as well. With that, we've also seen a massive increase in fast fashion. And in Australia, this is what our fast fashion waste looks like. So this is Australian data. In Australia, we purchase 28 kilos of textiles, new textiles per person per year, and 23 kilos get discarded to landfill. Now, what that means is an enormous amount of waste of resources, an enormous amount of pollution, which is impacting on humans and the environment, and now also economies. So many people on this Zoom will know that the globe is starting to realize the impact of the linear economy, focusing on a consumer's needs at the expense of all else. And I've got the source of these this diagram here. We're shifting to a circular economy, which is basically trying to design out waste and pollution at every step of the path, where we take resources, we make things, and we discard. So when we come to one of the grand visionaries of the circular economy, Jacqueline Kramer, Professor Jacqueline Kramer from the EU, from the Netherlands, she talks about hierarchical ways that we can approach materials. So the lowest thing we can do is recover the materials, so incinerate waste and recover it for energy. And the best thing we can do is to prevent a raw materials use. But there's a lot better things we can do since just recycling. So upcycling is good, downcycling is not the best. Value adding is much better than destroying things. So what I and the team that I collaborate with have been focusing on specifically this element of redesign at birth of the fibre. And I'm going to take you a bit further down that path. So in our work, my key collaborators and co-leads I'm showing here. This is the redesign. So redesigning fibres from birth. We know that cotton or plant fibres are natural and biodegradable, and we've been developing gene and foreign protein toolboxes because we know so much about the molecular components that make up that fibre as researchers, and producing natural and biodegradable fibres using GM approaches at the moment with the functionality of synthetics. We know that cotton has lots of great properties, but synthetics also have their benefits as well. What if we can create cotton that has that next step of evolution? So here's a paper that we published very recently, and we're very excited about this achievement. This was published just in December last year, so not very long ago. What this has demonstrated is that we've been able to use GM to transfer the beta lane pathway from the beetroot plant. That pathway does not exist within the genetics of the Malvalis, of which the cotton genus and cotton species is. That pathway doesn't exist naturally there. And we have engineered cotton plants to express this multi-gene pathway and paint or dye the fibres pink. So this is very exciting because it demonstrates that it's possible. It's not a perfect solution yet. We've got some technical things to fix the color and a lot of other things to explore, but that project is finished, and it really does prove that you can produce redesigned fibres from birth with novel properties. And that's the purpose of this work, is to design out waste and pollution. You can think about dyes, for example, or other non-biodegradable materials. And that's where we are really focusing at the moment. And with that, I will say thank you, and I shall stop sharing.
Thank you so much, Colleen. I think I speak for everyone when I say it's really fascinating research that you're doing, and we very much appreciate you taking the time to explain to us the biology of a cotton plant, and then also to explain a bit your research. I think sustainability is becoming a focal point of our industry, and so to see the work you and your team are doing to push that forward is really interesting for us. With that, I'd like to open it up to any questions that anybody might have for Colleen about her work or anything, her experience in the industry.
Hi, Colleen. Can everyone hear me okay?
Hello,
Okay,
Kim.
wonderful.
I can hear you. It's great to
good to
see
see
you.
you. I just want to say thank you for taking such a technical topic and explaining it to the rest of us ordinary people in language that we could completely understand. And I found it really very interesting on top of the sustainability that you're doing, which is amazing. But I think one of the biggest things that opened my eyes is to find out that a single fiber is a single cell. That has to be very, very unique. And is it one of the largest single cell? Okay,
Yes,
it is.
it is. So let me clarify. So absolutely, it is so extraordinary, and it hasn't become, the cotton fiber hasn't become a single cell just through natural evolution. It's because humans have been breeding it and breeding it to get longer and longer and longer. And our breeders are, around the world wanting to create longer fibers. So that's one of the reasons why it's so long. So it is one of the longest, if not the longest known plant single cells on the planet. There are animal cells that are longer, and you can think of an ostrich egg. People will say, biologists would say the ostrich egg is the biggest single cell probably on the planet. Yeah, but in terms of plants, definitely. And you can see it with a without a microscope. How cool is that?
Colleen, again, thank you for your presentation, and I also want to acknowledge the support that you gave to me when I started the Women in Cotton group and spoke to you in Brisbane 3 years ago, and also for Kim and I when we got on our journey with Women in Cotton. I have a question related to the work that you've done to produce pink cotton. Was there anything specific with the beetroot and the cotton that can be replicated with other colors? I mean, pink is obviously the best color to bring forward first, but is there anything biologically different for beetroot and cotton versus another color? Can we get blue? Can we get green? Are the other colors available too?
Excellent question. When the key person, so I had her photo up there, Dr. Madeline Mitchell. She was a postdoc who really was frontline in driving this research, and she was the one who wanted to go down the coloured cotton path. Her brilliance is extraordinary. Part of that, when we were going down that path, was looking at different types of colours. So that pathway can make orange, yellow as well. And through the GM stages, when we were making those genetically modified cells that could grow into cotton plants, the yellow transgenics never made it into little plantlets, and the orange ones wouldn't generate plantlets either. It's the deep purple ones who made it through to plantlets. So we could transform those embryo cells, but we couldn't get them all to grow into full, proper plants. So there's something about those pathways that might be toxic or lethal to the embryos when they try and change into leaves and roots and so forth. There are other potential pathways that could be explored, absolutely. Yeah, definitely.
Thank you.
I think Joseph had a question. He's raising his hand.
Yeah, a couple of them. Good morning, Dr. MacMillan. Going back to this chart about the formation of the fiber, I would like to ask you, how does the lack of water affects the length and formation of the cellulose wall? And another question about the circular economy. I suppose that, I mean, recycle, reusing implies that the clothes that are manufactured are 100% cotton, because otherwise it must be very difficult to separate cotton and synthetic fibers if you want to reuse it, right? That's the two questions.
Yes. Thank you for your question, Joseph, and lovely to meet you. So to the question of water and length, at the base of that initial cell that is going to elongate to make a fiber, there are pumps that will pump water in to drive turgor pressure, like when you fill up a water balloon. It's a bit similar to that. And so when there's not much water, you don't have much of that turgor pressure. And so it does change the rate at which it elongates, and it will change the quality of the fiber. Cotton plants are incredibly nuanced at how they manage water, incredibly clever and nuanced. I mean, it's a desert plant, so it has evolved to be very efficient with water. Sometimes in very dry years, depending on when the cotton plant gets water, at what stage of development of the fiber, you might even have finer, higher quality fiber sometimes, just depending on when the water was available. The other thing about water availability is it can be linked with heat stress, and if that occurs, sometimes you can have sterility of the pollen, which means you don't get fertilization, which means you don't get a seed, which means you don't get fiber being produced from that particular flower. So when it comes to the circular economy and cotton, 100% cotton, blends are probably the most complex thing to deal with in terms of waste. And I'm not just saying that in terms of textiles, but in terms of any area of the circular economy. Mixed plastics, for example, is extraordinary to try and deconstruct and then reconstruct. We are seeing some very clever science coming out to deconstruct cotton fibers with polyesters or other synthetic blends, so that through chemical processes, the cotton cellulose is degraded and it can be turned into a powder or other materials, leaving the synthetic fibers so they can be separated, and then they can go off into their more closed loop systems. So we're seeing some of that really nice tech starting to come through. I think your question is probably one of the most technically challenging one for the circular economy. And the other question that ties into that is using Industry 4.0 to help accelerate that in terms of identifying what is in this piece of fabric or this material, very quickly identify the chemistry, and then what path does it need to go down to deconstruct it, and then to keep the recycling process or the upcycling process going. I hope that's a useful set of answers for you, Joseph. Thank you.
I think Shoaib had a question as well.
Yes, thank you very much. And I would like to thank Dr. Colleen for her great research work And I appreciate that you're really doing great. So my question is actually to understand how practically this pink cotton is possible, means in terms of production of seeds or cultivation of this pink cotton, because it helps industry to get rid of dyes and colors and a lot of other, and which eventually contribute in sustainability and environment conservation. So I just wanted to learn in terms of its practicality and how it can go in a scale, means for industry to use.
Thanks so much, Shoaib, for your excellent question. You point to scalability, and that's one of the most beautiful things about a plant seed. Plant seeds are amazing. You can collect the seeds and plant the plant, and then the plant can generate its own seed and continue to propagate. So that's quite beautiful. In terms of this particular trait, this project is now over. It has ceased very, very recently. There is potential for the trait to continue. What Dr. Chao-Ching Lee and us as collaborators have not yet been able to test is fixing the colors so that it can go through all of the processes that fiber needs to go through before it becomes a yarn. So we need to make sure, with temperature, with water, with heat, whether we can make that. We haven't tested whether the color is stable and whether it will persist. So there's lots of practical things that need to be researched at an industrial level for that translation. But it's been proven it's possible. So the scalability piece, you point to the issue of dyes. That one singular issue in terms of water pollution, in terms of toxicity and human health for the workers who have to do the dyeing, and also in terms of waste management with toxic dyes. These are very complex things. So redesigning from birth is something that we should always be thinking about in terms of material production so that we can design out that pollution. So thank you for pointing to that too. I appreciate it.
And we had another question as well, Taslimal.
Thank you, Dr. Colleen, for your amazing presentation. Because when we are processing cotton fiber as a consumer in the manufacturing facilities, for us to know about this amazing research is quite, I really enjoyed it. Especially my question is quite related with Mr. Shoaib's one, because this pink cotton was very, very exciting for us also. And it's not just the scalability, the practicality issue that is the point you have been mentioning, that it is possible. Yes, we understand it is possible, but is it going to make any change in the manufacturing sphere in our area? And when you mentioned that your research on this is over, so my question was, what are the new areas that you are planning to research in future in CSIRO?
That's a great question. The future of this type of work, we're exploring options at the moment to see where we go next. We have particular market insights into what are the properties that brands and consumers are after, and the economics of that, and which ones we should focus on. But that's all I can say at the moment, because that's where we're at.
Thank you. Thank you so much.
I hope to see it progress and get bigger. There's much to be done. That's all I can say.
Colleen, I just wanted to talk about the science, as you're showing, is proving the concept and the possibility of producing coloured cotton, and also I'm assuming that there would be other traits that could be added to cotton in the future using similar research techniques. And the scalability or the production for commerciality, that's not necessarily in your space, but it would have to be a commercial entity seeking that knowledge and seeking to invest in that. Is that really where it would have to go up? Because I'm not really, your remit is to do the science, and then the market has to determine where that fits and how to invest further in it. Is that fair to say?
That is absolutely fair to say, and thank you for so eloquently articulating the remit of CSIRO. So it's an actual act. There's an Australian act, legislative act, which defines what CSIRO's remit is. And we are industrial and scientific research organisation. So partnering with the market to translate is critical, but that's not within my own bounds. So yeah. The translation piece is quite ripe, I would say.
I think you might be getting a lot of questions after this, maybe contacts of who do we go to for the investment and the commercial aspects of it.
Yes.
I think that's probably where we'll go. And just one more question on, if it were to be scalable, roughly how long does the research take to, make sure that the dye is stable, make sure that it actually can be done to scale? Would you have an idea of how long that would take?
It depends on the details of the specific question. A 5-year time scale just to look at those particular questions would be one answer. If we want to release a brand new variety, any variety, whether it's GM or whether it's natural breeding, it takes about, from very initial to very end, is 20 years. It's long-term scales. Sometimes we see GM varieties coming through faster than that. But the other side of the coin is the sooner we start, the sooner we'll get there.
Thanks, Colleen. Yeah, I think we looked at what we've done in Australia with the FibreMax seed, and I think we must be 20 years since that seed was introduced, and since it's gone around the world and benefited cotton growing in other countries as well. And that's part of the collaboration that you do in your area, as well as to reach out to other countries and who are engaging in similar research.
Are there any other questions or comments for Colleen and her very interesting research and topical for the cotton industry currently with sustainability? Well, Colleen, I think it goes without saying, but thank you so much for taking the time to go through your presentation today and to explain the cotton biology to all of us. I think that Kim was very correct in saying that you did a very eloquent job of explaining it to some of us who wouldn't necessarily know all of the science behind the fiber in the plant. So we really appreciate you taking the time.
You're welcome, and it's been an absolute pleasure to engage with everyone on the Zoom today. A lot of fun.
Just before we let you go, Colleen, I don't know, Katie, we haven't spoken about it, but there will probably be a lot of interest in this, either through this group when people go away and think about it, or through the recording as it goes up onto to the ICA YouTube next week. But is there a way that people can connect and ask questions and, put ideas forward, is there somewhere that they can contact and continue a conversation if that's possible?
For people on this call, they can contact my email address. I suspect it's on the slide there, which I showed at the end of the presentation. So you'll be able to see that there.
Okay, so if people have specific questions and they want to contact us, we can refer them to you, Colleen. Is that okay?
Yes, yes, that is okay. That is welcome and welcomed.
You will have 200 years worth of research by the end of these questions, I've got no doubt. Thank you, Colleen.
Yeah, thank you so much, Colleen. And I would just like to mention, everyone, thank you so much for taking the time to come out and listen to the presentation today. Our next Women in Cotton event is going to be a Chats for Change on April 6th. There will be more details to come out about that one soon, but please do mark your calendars for that date. And our next Women in Action event will be in May, where we'll spotlight another woman or two in the industry. So we really hope that you'll continue to join us and come back in April and May for our next events.
Thank you everyone for joining, and thank you again, Colleen, and we'll be talking in the next few days, no doubt.
Thank you. Bye-bye.
Thank you, Shoaib.
Thank you so much, everyone. Thanks, Colleen.
Thank you. Bye-bye.
Thanks, Joseph. Bye-bye.
Thank you so much. Bye-bye.
Thank you.